Welcome, welcome, welcome. It is Tuesday afternoon and therefore it's a little bit of biology revision. So today we are doing plant and animal cloning. This lesson has a particular OCRA focus. So this is what we're going to be going through this afternoon. We are going to be talking about natural plant clones, where this whole thing of plants reproducing themselves without any sex involved happens naturally. Then we're going to be talking about how we clone plants, how we do it as a result of cuttings and tissue culture. And we're going to do a little bit of evaluation, the pros and cons of cloning versus cloning versus sexual reproduction, and the advantage of cuttings against tissue culture, or doing a bit of a comparison at least. Then we're going to go and talk about the natural animal clones that occurs, how some animals do this asexual reproduction. And then we're going to talk about specific ways in which we clone animals artificially, by embryo cloning, or you might know it's embryo twinning, and somatic cell nuclear transfer, SCNT, that's dear old Dolly the sheep. And once again, we're going to go through a little bit of evaluation, pros and cons of the methods. So, hello, hi, lovely to see all of you. And yeah, really nice to have you joining me. So let's get on. And as always, we are going to start with a little bit of context. So here's your reminder. Clones are the offspring that come about as a result of asexual reproduction. And what's asexual reproduction? Well, A or at the beginning of a word means no, so it kind of means no sex. And just to point out that we can talk about cells cloning themselves. So mitosis is a cloning procedure because a parent cell gives rise to two daughter cells, two offspring, which are genetically identical. The two daughter cells are clones of the parent plant. But we can also talk about whole organisms carrying out cloning. So if you know, for example, plants which have got those long grass-like blade-like leaves, which are green and white stripes, they push out loads of tiny little plants. And that's why it's called a spider plant, because the little baby ones look like little spiders on their own stems. All those little spider plants are exactly the same, genetically identical to the parent plant. So, reminder about asexual reproduction. No fusion of gametes, no sperm, no egg, no pollen, no egg. If we're talking about plants, there is only one parent as a result of offspring of asexual reproduction. And the parent and all of the offspring are genetically identical, unless some kind of mutation has taken place. But there is certainly no variety as a result of the reproductive technique. So yeah, no variation. So natural cloning, so it happening naturally in nature, is really advantageous if the environment is favorable and stable. So if it's favorable and a stable environment, and for some reason, the opposite sex isn't around, then asexual reproduction is a fantastic way to go for organisms, because it means they can colonize the favorable environment. And there is no need to bother with the production of gametes, the fusion of gametes, and possibly the nurturing of the embryo, particularly for animals, obviously. And that's fine as long as the environment doesn't change. But of course, environments change kind of all the time. And that's where sexual reproduction kind of comes into its own, because sexual reproduction brings about stupid amounts of variety. as a result of meiosis and as a result of the completely random fusing of the sperm and the egg, or the pollen and the egg. And that variety is absolutely brilliant when environments change, because, of course, if the environment changes and even most of the population is wiped out, it's quite possible that just a few individuals will be able to survive and reproduce, passing on those favourable alleles to their offspring, bloody, bloody, bloody. Okay. Lavinia, is this lesson for CIE? I'm going to be really honest with you. You're going to have to check your specification. It should be really clear in your specification. I haven't done that today, if I'm honest, to check whether this cloning, plant and animal cloning, is in CIE. I'm afraid you're going to have to check that. And I don't know off the top of my head. I'm sorry. Okay, so now let's go on to talk about natural plant cloning. How plants do it naturally of their own accord without any interference from humans. And notice that it always involves the stems. So you just need to be aware that strawberry plants, you might be aware that strawberry plants send out runners, stems that run above the ground. Another word for them is stolons. Personally, I prefer runners. And then produce a little tiny plant at the end, which then has its own roots, and the stem between the parent plant and the baby just kind of dies, and you get the new plant. You have suckers, which are stems that come out of roots. So here you can see the main trunk of the tree, hopefully, but there's a whole load of suckers that have come out of those roots. And again, those suckers, eventually the roots that join the main stem and the sucker would die, thereby bringing about a new independent plant. Bulbs, underground stems. Who knew? Well, you can tell that bulbs are underground stems, so think garlic, onion, and if you know any flowers, think things like bluebells or daffodils. The stems, can you see that the roots are on the bottom, and therefore the bit that is doing the whole layer and swelling thing is actually the stem. And what happens here is that when a bulb gets particularly big, a little tiny one is produced on the side, and that gradually gets bigger and eventually becomes separate to the parent plant, genetically identical. Tubers, potatoes. You would have thought that potatoes were kind of swollen bits of roots, but from a kind of anatomical point of view of the plant, they're actually underground stems. And finally, here's another example, underground stems. They're called rhizomes. So runners go, kind of run above the ground. Rhizomes are the kind of ones that are sent out underground. So rhizomes, these happen in flowers like irises and lilies, and I'm no gardener, so I'm running out of examples, but there we go. Loads of ways in which plants naturally reproduce themselves asexually. No requirement for eggs and pollen to meet and fuse. Notice no seeds involved. If plants produce seeds, as soon as you see the word seeds, that is sexual reproduction, because a seed is kind of the equivalent of an animal embryo. It grows, it develops, the cells differentiate into roots and shoots, you get an adult, fully differentiated plant. So always bear that in mind, that if you see seeds in a question or in any bit of blurb, it means that sexual reproduction has come about in order to produce the seed. Okay, so now we go on to plant cloning techniques. So shouldn't have done that. So, number one, taking cuttings. So I am now about to give you a mark scheme method for taking cuttings. So first of all, you cut the stem. The stem is the most common part of the plant that is used for cuttings. And notice, and here's a little diagram, that you cut it at a slant and you take the cutting from a healthy plant. This sentence, without even the bit in the brackets, are 2 marks on the mark scheme for OCRA. Cutting a stem from a healthy plant, 1 mark. Cutting it at a slant, 2nd mark. If you dip the lower end into rooting hormone, that would give you your 3rd mark. The rooting hormones cause cell differentiation. So what you had is a stick of a stem, but from the base of the stem, the cells will redifferentiate into root cells, becoming roots, obviously. And finally, you place that into watered compost. Ignore this diagram where it talks about the sand. Nothing to do with anything, not going to get you a mark, but I just like this nice clear diagram of basically the simple technique of cutting off a bit of the plant, healthy plant, dipping it into rooting compound, putting it into nice watered compost. That would earn you 4 marks in the mark scheme. Worth noting that the other 2 possible marks were, and can I now remember them? Yes, you cut the stem between 2 nodes. I personally thought that was a detail that I'd be less likely to remember, so I didn't put it in. And the final possible mark was at the end, if you put a piece of plastic, covered the plant with plastic to reduce transpiration. And again, I thought that was a less obvious mark. But this is your 4 mark scheme answer for taking a cutting. Hopefully that was useful. Okay, so tissue culture. This is about producing hundreds of little plants, and it can be called micro propagation. It is a whole lot more complex. It requires a load more complicated equipment and scientific know-how. So notice all the terminology that I'm using here. First of all, sterilized. Sterilize, you are cutting little parts of the plant, small pieces of meristematic tissue. Meristematic, remember, is that undifferentiated bits of tissue, often at the tip of the root, tip of the shoot, tip of the root. There is also some meristematic tissue in the stem, but you're basically looking to get small pieces of meristematic tissue. You sterilize them with ethanol. This happens to be little bits of stem of sugarcane, these pictures are. B and C shows that when you then put them onto the sterile medium, this brings about callus formation. And the callus is a mass of undifferentiated cells. And you can hopefully see those undifferentiated cells growing. D and E, those calluses, I think that's right, those calluses are then moved onto different media with different hormones and of different concentrations in the media. And that brings about the root differentiation and the shoot differentiation. And then finally, when the little plantlet has its roots and shoots, in other words, it's pretty much fully differentiated, it can then be planted into watered compost for the plant to grow independently, as it were. Okay, so a little bit of evaluation for the plant cloning. So in terms of cloning, plant cloning, in other words, asexual reproduction of plants versus sexual reproduction of plants, notice I've reminded you here that that involves seeds. Cloning, asexual reproduction by either cuttings is the really most obvious one. Well, certainly at a kind of domestic level, one that you can do at home. It's way faster than seeds. And you don't need any gamete fusion. You're almost growing a plant from a piece of stem, which is just more, it's less vulnerable than a seed, which has to go through germination and then development. All of the plants that you clone have the desirable characteristics because they're going to be genetically identical. Notice I've picked out mark points here because also you can say that they are uniform, and that means that the plants are easier to grow, to transport, and to harvest. But the downside is that if they grow faster, you're more likely to have overcrowding. And here, notice that these two connect because they all have the desirable characteristics and they're all uniform because there's no variation. And therefore, they are going to be susceptible to a new disease. Now, cuttings versus tissue culture, let's do a quick comparison of those. Cuttings require far less expensive and complicated equipment. You basically need a very sharp knife and a bit of rooting powder. That's it, rooting hormone. Cuttings require less scientific expertise. So, for example, cuttings really don't require any form of aseptic technique, whereas tissue culture, that micropropagation, absolutely does. Contamination would overrun the developing calluses, for example. Cuttings produce far less offspring. If you can get micropropagation going, you could have hundreds of plants, not just one or two, which would kind of come from cuttings. Cuttings are faster than tissue culture. Okay, Johnny. Oh my goodness. Right, Johnny, with regards to, just quickly, seeing as we've just come to the end of plant cloning, in terms of you trying to ace your A-level physics, I'm going to be really honest with you. I am not going to be somebody who's brilliant to ask. I personally know some fantastic A-level physicists, but they're not tutors as far as I know. And I don't know of an A-level physics teacher who is doing this kind of thing on YouTube. So I'm really sorry, Johnny. I don't think I'm going to be able to help you. Manpreet, no, this isn't AQA. This is OCRA. This is an OCRA specialist piece of the specification. So I hope that is helpful. I'm sorry, it isn't. And is it for SNAB? You are going to, I know it's not for AQA because I know the AQA A-level spec inside out and back to front. You're going to have to check your spec, do a quick check on your phone. And I do Control F to search the document and just put in plant cloning or something like that. So to see whether you need to know that. I hope that helped. Sorry, Manpreet. Okay, so just really quickly, just to say, I have a website, and it practically, I mean, I'm up to about 98% of covering the whole of AQA and the whole of OCRA syllabuses for A-level, both years. And if you want to get 2 years worth of videos, in other words, the whole flipping course, you can have it for 50 quid. It's half price. On the website, it says 100 quid. In the description, it tells you the code to put in. I mean, there's nothing hidden here. So get it for half price for the whole year. It could even still be worth it because 50 quid is kind of about 1 hour of private tuition, and you get everything. And every video has me going through exam questions at the end. If you just need AS, £25 for the year. And this is me trying really hard to make this accessible to every flipping student, regardless of family income. If you can get 10% commission, if you recommend it to friends and they buy it using a link that I send you, you get 10% commission. You could even make a flipping profit. And there's a freebie, so do just go and have a look at that, if nothing else. So now let's go on to natural animal cloning. Now, actually, I almost think my notes are the wrong way round here, because the first thing I'm going to talk about is the fact that identical twins are an example of natural human cloning. Identical twins are human clones. Now, here's the thing, though. Obviously, human twins have come about as a result of sexual reproduction. Yeah, the sperm and the egg are fused. But here's the thing, the zygote, or really early embryo, all right, that's all a zygote, it's really early embryo, it actually splits to form two embryos that then develop to become two genetically identical humans. But here's the weird thing to get your head around. Actually, the embryo is the biological parent of the two identical twins, because they are clones of that embryo. They're not clones of their mum and their dad, which are obviously their biological parents. Yeah, but in terms of, we talk about humans being, identical twins being human clones, they are, but they're clones of the parent embryo that did the splitting. Because in effect, you've got two identical offspring from a single parent. I hope that makes sense. We're going to talk about embryo twinning, and it will be even more obvious then. So, example of identical twins. So here are some other animals that do natural. Here's the weird thing. These animals, from a single parent, they produce loads of offspring with no sex going on, no fusion of gametes at all. So aphids can produce hundreds of themselves by asexual reproduction. Bees and wasps, some bees and wasps can do the same. Komodo dragons, those giant lizards, can do it. And some sharks, namely the zebra shark, produces genetically identical offspring. But the zebra shark, if there aren't mates around, the female will produce a whole load of genetically identical offspring, loads more females. The Komodo dragon, here's one that's going to blow your brains. The Komodo dragon, the female asexually produces males. So clearly they're genetically identical, except that it has the sex chromosomes for the male. Anyway, get your head around that. Or weird. Obviously not all straightforward biology. Very interesting though. Anyway, so artificial animal cloning. I'm going to try. That's better. Did that better this time. So artificial animal cloning, we're going to talk about embryo twinning. OTRA call this embryo twinning. I would call it embryo cloning. So here we go. Here's the nice big diagram that explains what's going on. There's not much point in me writing more if a diagram does the job. Notice that the embryo, which is produced by sexual reproduction, this is the parent. So because the cells of the embryo get separated, they're allowed to develop into embryos. The embryos are put inside surrogate mothers, and they then produce offspring that are identical to the embryo, the original embryo at the top of the diagram. Notice that these offspring are not going to be identical to the mother and father that provided the sperm and the egg. They're only identical to this embryo, and therefore the embryo is the parent of this asexual reproduction. The embryo, this original embryo, can be produced in vivo, in other words, inside the animal, or it can be produced in vitro, in other words, in a petri dish in the lab. All right. This bit all happens in vitro. You've either got the embryo in a petri dish and you then separate it, or you take the embryo from the uterus of the animal and then separate the cells in the lab and then allow them to develop into embryos and put them into surrogate mothers, so into the uterus. So that's how embryo cloning or embryo twinning works. The second way, which you need to know how animal cloning can happen, is somatic cell nuclear transfer. Bit of a mouthful, so let's try and work our way through here. So notice that we often do animal cloning of farm animals, i.e. animals that are economically important to us. They're the ones that provide us with the food. That's why all of these examples are of cows and sheep, because let's face it, those are the main mammals that we get our food from, whether it be meat or the dairy. Okay, so here we have sheep A and here we have sheep B, very different looking sheep. From sheep A, we are going to take, let me just, Johnny, sorry, just realizing all the questions. Johnny, yes, I do. I have sample videos on YouTube. Got loads of them on YouTube. There's quite a lot of live streams that I do on YouTube. I have also got properly edited videos. Classic ones that seem to be very popular are my immunity ones, my respiration ones, and my photosynthesis ones that seem to just clarify the process for things. And I absolutely cover AQA on my website. Yes, I do. Do go and have a look. But yes, the sample ones are on YouTube. And I think if anything, you know, kind of as I've gone along, the videos have got better. I've got better editing and all that kind of stuff, as you can imagine. This is, yes, Kauza. I hope I've said the name right. This is how somatic cell nuclear transfer is how Dolly the sheep was made. Yes, I should have said that right at the start. Dear old Dolly. Yeah, so there we go. So now now that we've established that this is Dolly the sheep territory, let's work out what on earth went on to make Dolly the sheep. The most dramatic thing, the most significant thing about Dolly the sheep being produced, and I think she was made, late 90s, so way before your time. It was the first time that a mammal was produced from an adult, fully differentiated cell. And this is how it worked. So scientists, and they were British scientists, sorry, just doing a little bit of patriotism here, British scientists up in Edinburgh in Scotland, Scottish scientists. They took a somatic, and all a somatic cell is a normal body cell. But notice, any body cell is going to be fully differentiated for its job. They happened to take a skin cell and they took it from the mammary gland of the sheep, the breast of the sheep, to you and I. So they took the body cell, fully differentiated skin cell, and from another sheep, they took an egg cell. They removed the nucleus from the skin cell, the somatic cell. Somatic cell nuclear transfer. Hopefully this is starting to make sense. And the egg cell, so the nucleus was removed. And the egg, the nucleus was also removed, except this nucleus was chucked in the bin. And we now can call this egg an enucleated egg, and it's just quite a useful term to use if you're having to describe the process. The enucleated egg is then fused with the nucleus of the somatic cell by electrofusion. You only just have to know the word electrofusion. It just means that a tiny pulse of electricity, which caused the nucleus and the cytoplasm of the egg cell to fuse. This then was allowed to develop into an embryo, and the embryo was then implanted into a third sheep's uterus. Okay? And this gave rise to a lamb, Dolly. Hopefully it's apparent that it makes sense that Dolly was genetically identical to sheep A, because sheep A was the sheep that provided the genetic information, provided the nucleus. So that is somatic cell nuclear transfer. The nucleus from a somatic cell being fused with an empty egg cell. That cell then being allowed to develop into an embryo, undergo mitosis, develop into an embryo, implanted into an adult sheep, into another surrogate mother, and producing a lamb that's identical to the sheep that donated the nucleus. Okay, so evaluation, we're nearly there. So artificial animal cloning, useful. Now here's the thing, think of any situation where an animal, there are very few of them. So endangered animals, funnily enough, extinct animals, if you can recover the nucleus or recover all the DNA, or if you knew an animal was going to go extinct and you therefore froze the eggs so that you could recover the nucleus, yeah? But also notice that this allows for pets to be cloned. We could have a whole discussion about whether that is sensible or not, but yeah, it allows pets to be cloned. It allows genetically modified animals to be cloned. You know, genetically modified animals are not going to be your run-of-the-mill animals. They might be in very short supply. So, for example, an animal that has been genetically modified to produce a particular medicine protein in its milk, you might choose to genetically modify that animal because it's taken such a lot of expertise and time and money to produce it in the first place. But also notice that we, again, use this to increase the population of elite farm animals, you know, the ones that we use for food. And elite just means the best examples that are either particularly meaty or particularly have a particularly high milk yield, obviously something that is useful to us. All of those animals, notice, you will have all of the desirable characteristics in the clone. And here's another point, is that cloning can happen at any time of the year because, of course, there are no breeding seasons for this process. And here's the slightly contentious one, and I'll talk about this a bit more when I talk about the disadvantages. We can produce a clone in order to produce stem cells, genetically identical stem cells, tissues and organs to the parent animal. So, not surprisingly, this animal cloning requires expensive equipment. It requires scientific expertise. We are not talking the same kind of way that we can take a cutting. Yeah, it's just way more complex than that. No genetic variation. Notice how this links. You know, they all have the desirable characteristics, but they are all exactly the same. And again, we have that whole susceptibility thing to either a massive change in the environment or new disease or pests, for example. And notice that this links with this one because this is about the cloning of human embryos. So, you know, ethically, it has been agreed that human embryos can be cloned up until, now I think I'm right in saying, for just the first 14 days of development of the embryo. The embryo is not allowed to develop further than that. So it is really only a ball of cells, doesn't have a nervous system, a heart, any of those organs. But clearly, an awful lot of people would see that as highly unethical because they would absolutely believe that life started at conception and would therefore find this idea entirely unacceptable. But this is where potentially you could get stem cells. And of course, if you get stem cells from an embryo, you are obviously potentially damaging or destroying the embryo. So there's a whole ethical situation there. But hopefully that has helped you. And here we go. Oh, we've got a load more questions. Hopefully that has helped you. I'm just going to consolidate what we have done, and then I'm going to go through the questions that have come up in the last few minutes. So hopefully we have talked about what clones are. We've talked about examples of natural plant cloning and natural animal cloning. but we've really focused on how we clone plants and how we clone animals for our own advantage. Plant cloning with cuttings and tissue culture, animal cloning with embryo twinning, and with somatic cell nuclear transfer, Dolly the sheep. So I'm hoping, and we've obviously also done the evaluations, pros and cons, and comparing the different processes. So I'm really hoping that that has been useful. But now let me just see if there are any questions. Okay, could they use sperm cells nucleus? They couldn't use a sperm cell nucleus for any form of animal cloning, because of course a sperm cell only has half of the DNA in it. You need to have all of the DNA. Remember that the egg cell has that cell removed. The cytoplasm of the egg is kind of there because it's kind of programmed to divide, yeah, to become an embryo. Hopefully I've explained that now, Kausa. Thank you, Kausa. Johnny, can you tell me where the freebies are? If you go onto the website, it should be, the landing page should give you the three main things that are on offer. The freebie, how to increase your grade up to an A star, the AS bundle of videos for 25 quid for the year, and the year 1 and year 2 bundle of videos, which is 50 quid for the year. Yeah, absolutely. I completely understand you wanting to see the videos. If you want to see what a video, one of my normal teaching videos looks like, go onto YouTube, because I've actually kept some of my most popular ones on YouTube. So as I say, you know, immunity, respiration, photosynthesis. So have a look at those. They'll give you a good idea. And they're quite old hat ones, I think. I've got their stuff, but anyway. Oh, what do I think about human cloning? I personally, I would be, yes, absolutely not interested. Having one of me in the world would be bad enough, let alone having multiple copies. What an awful thought. Imagine having more than one of each of your teachers around. That's not great. You know, the whole thing that your clone would be a different generation to you, potentially. Oh, very weird. No, no, not at all. Johnny, for the freebie, it won't need you to pay. It won't need you to pay. Everybody, I've had hundreds of people pick up the freebie. So do feel free to message me in the comments of this video, and we can sort out the problem. And are all my videos basically the style of what I've done in this live stream? Fundamentally, I tell you what I'm going to cover. I go through put the whole lesson in context, and then I really focus on what is required by your exam board. So that's what I try and do. Yeah, okay. I don't know what you mean. Via the screen. Can I do this? I don't know. Let me just see if I can. I'm not sure I'm going to do this because I don't particularly want to show what's on my laptop. I'm actually going to say no, Johnny, but I will put a link in the comment section of this video on YouTube. I hope that helps, Johnny. All right. I'm on Instagram, flipping DM me. All right, so we'll sort it out. We'll sort it out. Okay, all right. Kausa, right. What if there's a person with a rare genetic mutation that gives them a big advantage and you clone them for the better of mankind? That's a tricky one. Do you know, I think by far a more tricky one is actually, oh yeah, no, okay, yeah, or we have people that without disease, or you clone somebody so that they could give their clone a kidney transplant or something. Oh, it's all, do you know, I think it is an absolute ethical minefield. You know, talk about, you know, every cliche, thin end of the wedge, slippery slope. You do something because initially you think it's great and it works, and then you suddenly realize the huge consequences for doing something. And I think fundamentally, I think I'm all for every single human being unique and being of equal value. How does that work with a, oh my goodness, you're absolute minefield, absolute flipping minefield. I'm not doing any more of this. Okay, I hope that's useful, and look forward to seeing you next week. I'm going to be going through paper 1 2018 OCRA paper next week, so hopefully that will be useful. All the very best. Have a lovely Easter weekend. Whether or not you celebrate Easter, have a great break, and I will see you next Tuesday. Bye-bye.